Okay, 7:30. Good morning. It's my and welcome to the Grand Rounds presentation by Dennis Wang, the outgoing recipient of the Bruce Laughlin Endowed Fellowship in Cardiology. It's also my pleasure to announce this year's recipient, Dr. Sateh Shakil. If we can do virtual applause. The Bruce Laughlin Fellowship was established 15 years ago by the Laughlin family to honor their father, Dr. Robert Bruce, the first head of the Division of Cardiology at the University of Washington. Dr. Bruce was the quintessential academic cardiologist. He was an imaginative and highly focused clinical investigator whose special contributions deserve high ranking in any list of important medical milestones. He's best remembered for the Bruce multi-stage treadmill test, which by its simplicity of design became a reproducible, readily performed estimate of maximal exercise capacity and by extension, maximal oxygen consumption. I'd also like to take this opportunity to thank the whole Laughlin family who have continued their generous support of division fellows and faculty over many years. They are incredibly kind and gracious, embodying the best characteristics of their father, Dr. Bruce. Now on to Dennis. Dennis graduated cum laude in 2003 from the University of California, Berkeley, with a double major in computer science and molecular and cellular biology. He then completed an MD PhD at the University of California, Irvine, receiving multiple merit-based scholarships and awards. During his PhD training, Dennis focused on the discovery of a mitochondrial protein complex that fine-tunes oxidative phosphorylation through regulation of mitochondrial mRNA poly-A tails. Dennis then did his internal medicine training at Baylor College of Medicine. Since coming to the University of Washington in July of 2016, Dennis has distinguished himself, demonstrating creativity in overcoming both basic and clinical research hurdles, as well as energy in applying for and successfully attaining research funding. This includes an American Heart Association postdoctoral fellowship award, as well as several institutional grants, including a Locke Foundation award. In collaboration with Dr. Rong Tian, director of the UW Mitochondrial and Metabolism Center, and myself, Dennis has focused on a completely novel approach to the treatment of heart failure by therapeutically targeting mitochondrial dysfunction. Dennis will describe this work today. In closing, I also should mention that Dennis has a revised manuscript under consideration at the Journal of Clinical Investigation, where, not surprisingly, Dr. Bruce published his first description of the concepts underpinning the development of cardiac exercise testing in 1949. Dennis?
Good morning. Thank you, Dr. O'Brien, for the warm introduction. I would like to thank you all for joining us today, particularly the Laughlin family. Welcome. I have no relevant disclosures. Today, I'm very excited to have the opportunity to share my research project with you. We'll start by discussing the background of NAD. We will then examine how boosting NAD in the heart affects cardiomyopathy and NAD's potential anti-inflammatory effect in heart failure. Lastly, we'll go over a number of related human heart failure trials. I thought I would start by explaining how the scientific community became interested in NAD. I'm sure you're all familiar with the concept of the fountain of youth. For thousands of years, human beings of all cultures have been seeking for ways to stay young, usually in the form of a mystical fountain to bathe in or a drinkable potion. As illustrated in this painting, Qin Shi Huang, the ruler of China around the 3rd century BC, is famously known for sending a convoy consisted of 3,000 young men and 3,000 virgins out to the East Sea to look for the magical potion that will allow him to live forever. By some accounts, this convoy reached the island of Kyushu, Japan, and ended up staying because this potion was nowhere to be found. Though time has passed, our urge to stay young and healthy has not changed. Over the past 20 years, there have been numerous accounts documenting the association of aging and NAD depletion in various organs and tissue types, particularly in animal models, but also in humans. Taking the rat as an example, which has a lifespan of about 2 years, the heart of a 20-month-old, equivalent to 80 years in human age, has about a 30 to 40% decrease in NAD than that of a 3-month-old heart. These observations led to the notion that NAD depletion is a fundamental feature of aging, and any agent that restores NAD may potentially be the long-sought youth potion. NAD, as many of you remember from college biology, is the carrier that donates high energy electrons to the electron transport chain to generate ATP. Via catabolic pathways such as the TCA cycle, beta oxidation, or glycolysis, the potential energy stored in carbon-carbon bonds of glucose and fatty acids get converted to reducing potentials by transferring electrons to NAD+ to form NADH. And NADH can be oxidized or consumed by the electron transport chain to pump protons for the mitochondrial matrix into the intermembrane space. This electrochemical gradient is what allows complex 5 or ATP synthase to generate ATP. The electrons are ultimately accepted by oxygen to form water. In addition to its role as a high-energy electron carrier in oxidative phosphorylation, NAD+ is a coenzyme for a number of important cellular activities. I want to focus on the sirtuins, which is a family of NAD-dependent deacetylases that have been shown to regulate mitochondrial function, circadian rhythm, cell cycle, and inflammation. The typical deacetylation reaction is shown on the right. As you may recall, lysine residues can be acetylated as part of a protein's post-translational modification. A protein may have multiple acetylation sites, and the acetylation patterns often dictate the biochemical properties and the enzymatic functions of the protein. Sirtuins, on the other hand, can remove acetyl groups from the lysine residues in response to certain cellular signals. In this process, NAD+ is consumed to become nicotinamide, or NAM. Here's a busy slide that illustrates the various metabolic pathways for the biosynthesis of NAD. The de novo and the Press-Handler pathways use tryptophan and nicotinic acid respectively to generate NAD. Concurrently, there is a salvage pathway named after the fact that it involves recycling of the byproducts of NAD-consuming enzymes, such as sirtuins, back to NAD. NAM, nicotinamide, as we alluded to in the previous slide, can be converted back to NAD via one intermediate called nicotinamide mononucleotide, or NMN. Another important molecule worth mentioning here is nicotinamide riboside, which is an FDA-approved dietary supplement available as an oral tablet. Now, nicotinamide riboside, or NR, can be converted to NAD via 2 enzymatic steps. In an open-label pharmacokinetic study targeting healthy individuals, Dr. Sophia Earhart and Kevin O'Brien demonstrated that 8 days of NR oral administration at a final dose of 2 grams per day resulted in roughly a 2-fold increase in whole blood NAD levels. In this study, NR was very well tolerated, and the study subjects were free of severe adverse events. In addition to this endeavor, there have been 2 additional studies that also showed that in healthy subjects, NR administration results in consistent elevation of whole blood NAD levels and is safe and well tolerated. Earlier this year, a Japanese group studied the safety and pharmacokinetics of single-dose NMN administration at 100, 250, and 500 milligram in 10 healthy individuals. Similar to NR, NMN administration was safe and free of adverse side effects. Unfortunately, the group didn't report any change of the whole blood NAD levels. Instead, they showed a dose-dependent elevation of NAD metabolites over 6 hours after the drug was given. Now I'm going to shift gears and talk about NAD and heart failure. There has been tremendous interest in the heart failure community in targeting the mitochondria as a potential therapeutic for many years. The aspiration stems from the observations that the heart is highly metabolic and produces and consumes kilograms of ATP on a daily basis. To achieve this, the mitochondria is estimated to make up about 40% of the heart by weight. The preferred fuel source of the normal heart is fatty acids. In contrast, failing hearts have been shown to be metabolically dysfunctional and switches to use glucose and ketone bodies as food source. Some compare this phenomenon of glucose switching to the Warburg effect in cancer. What these observations imply is that a failing heart undergoes a fundamental metabolic rewiring to accommodate the energy imbalance where the demand exceeds the supply. As all of you are aware, the current guideline-directed medical therapy for heart failure tends to focus on reducing energy demand. For example, beta blocker reduces heart rate, ACE and ARB reduce afterload. The question we mitochondrial people, if you would like to ask is whether increasing myocardial energy supply by improving mitochondrial function may be beneficial in heart failure. Specifically, can we achieve this by boosting NAD? Over the past 10 years, there have been several groups around the world who have been asking the same question using various animal models of cardiomyopathy. In 2013, a former postdoc in our lab demonstrated that mice with mitochondrial dysfunction have a propensity for acquiring cardiomyopathy. In this study, a genetically modified mouse strain with complex 1 deficiency was used, resulting in severe mitochondrial dysfunction. Now, without extrinsic stress, despite the mitochondrial dysfunction, the knockout mouse hearts exhibited comparable LV function and dimensions. The lifespan was also comparable to control mice. However, when a stress is introduced, by subjecting the animals to a transverse aortic constriction or TAC surgery, which essentially ties down the ascending aorta to artificially increase afterload, over a matter of weeks, the complex 1 deficient mice exhibited worse cardiac function, LV dilation, and increased heart rate. In a follow-up study using the same complex 1 deficient mouse model, another postdoc in our lab showed that myocardial NADH to NAD plus ratio was elevated with induced cardiomyopathy. This is likely due to a decrease of NAD plus level. Now, this can be partially restored with concurrent treatment of NMN. And remember, NMN can be converted to NAD via one enzymatic step. At the same time, the mitochondrial proteins in the heart have been shown to be hyperacetylated in the knockout mice subjected to TAC, which can also be partially restored with concurrent NMN treatment. Importantly, NMN treatment could also partially restore the LV function by fractional shortening here. In summary, our current working model states that when a vulnerable individual encounters a cardiac insult, as in the example of TAC being applied to a mitochondrial deficient animal, the NADH to NAD plus ratio go up, again, likely due to a decrease of NAD plus, which results in hyperacetylation of mitochondrial proteins and subsequently contributes to the cardiomyopathy phenotype. Now, in this model, if we're able to correct the NADH to NAD plus ratio by boosting NAD, say by giving NR or NMN, we may be able to reduce protein acetylation, possibly by a sirtuin, and ultimately lead to recovery. I would like to discuss another mouse model of induced cardiomyopathy where boosting NAD was shown to be cardioprotective. In 2018, a French group reported a mouse model of dilated cardiomyopathy triggered by cardiac-specific deletion of serum response factor, SRF. Now, SRF is a major transcriptional regulator of genes and microRNAs involved in energy flux and calcium signaling, ultimately affecting contractility. Using this model, the group showed that SRF depletion resulted in a dilated cardiomyopathy phenotype. And consistent myocardial NAD depletion. This time, the group used NR rather than M to boost NAD. Again, NR can be converted to NAD in two enzymatic steps. As illustrated here, NR supplementation resulted in restoration of myocardial NAD level, as well as improved LV function and LV chamber size. So far, we have covered a number of animal cardiomyopathy studies that demonstrated the cardioprotective effect of raising NAD at the level of the heart. However, when NR or NMN is given, the effect is in fact systemic, one of which we believe is an anti-inflammatory effect. There's little doubt that heart failure is an inflammatory condition. The cardiology community has long observed that heart failure is associated with elevated clinical biomarkers of inflammation, including ESR and CRP, as well as circulating inflammatory cytokines, such as IL-1 beta, IL-6, and TNF alpha, based on a series of studies in the 1990s. In addition, in this multi-center clinical trial of 1,200 patients, Dr. Nita Deswell and her colleagues demonstrated that elevated levels of TNF alpha and IL-6 are independent predictors of mortality in patients with advanced heart failure. Here are the Kaplan-Meier curves of advanced heart failure patients broken down into 4 groups by their circulating cytokine levels. As you can see, patients in the top quartile, denoted by these solid black lines here, are associated with the worst outcome. Dr. Deswell happens to be one of my mentors when I was an internal medicine resident at Baylor in Houston. In addition to her scientific excellence, she was also a spectacular cardiologist. I think she even wrote one of my recommendation letters for the fellowship application. I'm very proud to be presenting her work during my Grim Rounds presentation today. Now, around the same time, in a series of animal studies, it was demonstrated that introduction of cytokines, such as TNF alpha, resulted in diminished myocardial contractility. These experiments are performed with explanted animal hearts, subject to the Landorf preparation, where a balloon is placed inside the left ventricle for pressure measurements while the heart is beating inside a bath of warm physiologic buffer. The change of LV systolic pressure, or simply the left ventricular diastolic pressure, are surrogate markers for myocardial contractility. Observations we just went over led to the model of sterile inflammation in heart failure. By this model, myocardial injury or damage due to, say, MI or viral myocarditis or simply chronic heart failure results in the leakage of mitochondrial contents, including mitochondrial DNA, RNA, proteins, peptides, phospholipids, and so forth, into the circulation. The collective term for these mitochondrial content is mitochondrial damage-associated molecular patterns, or mitodamp. Now, these highly immunogenic molecules activate circulating immune cells via pattern recognition receptors to produce cytokines. And these cytokines can circulate back to the heart to harm the heart even more, resulting in a vicious cycle. It was observed early on that the mitochondria is often dysfunctional in circulating immune cell activation. In our manuscript currently being reviewed at JCI, we collected blood samples from 19 hospitalized stage D heart failure patients and 19 healthy control subjects, and isolated their circulating monocytes and lymphocytes, collectively called peripheral blood mononuclear cells, or PBMCs. So from this point forward, whenever I mention PBMCs, I'm referring to the combined circulating monocytes and lymphocytes. What we observed was that the basal oxygen consumption rate of the PBMCs did not differ significantly between heart failure and healthy. However, the maximal respiration, which is measured in the presence of a chemical that obliterates the mitochondrial proton gradient, allowing the electrons to flow unopposed down the electron transport chain, is significantly lower in the heart failure group. At the same time, the inflammatory cytokine gene expressions were found to be significantly elevated in heart failure. We were able to recapitulate this phenomenon in vitro. Patients undergoing LVAD surgeries, I was able to procure pieces of their myocardial tissues and isolate the mitochondria. I subsequently lysed the purified mitochondria, spun down the soup, and took the supernatant containing mitochondrial DNA, RNA, proteins, and so forth. I call that mitoDEMP. Treating PBMCs from healthy subjects with mitoDEMP resulted in a decrease of maximal oxygen consumption, as well as a surge of cytokine gene expressions, similar to what was observed in the heart failure PBMCs. And however, as you can see, what we're working with is a hodgepodge of mitochondrial content, which is difficult to quantitate. To simplify our model, we decided to focus on mitochondrial DNA as our prototypical mitoDEMP. Now, based on work published in the past 10 years, mitochondrial DNA can be either actively secreted or passively leaked into the circulation in the setting of cellular stress or tissue necrosis. A Norwegian group demonstrated 4 years ago that heart failure is associated with elevated blood plasma levels relative to healthy controls, as well as by NYHA functional class. In this model, mitochondrial DNA activates pattern recognition receptors such as toll-like receptor 9, TLR9, to trigger downstream cellular cascade, including activation of transcriptional factor NF-kappa B to turn on the gene expressions of pro-inflammatory cytokines, as well as induction of mitochondrial dysfunction and mitochondrial ROS production to facilitate the maturation and secretion of the cytokines. Now, TLR9 recognizes unmethylated CpG motifs, which are often found in mitochondrial DNA and bacterial DNA. CpG motifs is simply a 5' to 3' CG sequence in any part of the DNA molecule. The P denotes the phosphate group that bridges the two bases. Again, mitochondrial and bacterial DNA are often unmethylated at the cytosine residue. In comparison, most, if not all, nuclear DNA are methylated at the CpG motifs. From an immunologic perspective, this is one way for the body to tell self from non-self. I recently came across an interesting manuscript that showed treating circulating immune cells with a 22 nucleotide DNA oligo containing repeated unmethylated CpG motifs resulted in the cell's release of mitochondrial DNA. As you can see, within minutes of treatment, mitochondrial DNA can be detected in the extracellular space. What this implies is that circulating immune cells have the ability to detect DNA with unmethylated CpG and respond by secreting mitochondrial DNA. This study gave us the inspiration that mitochondrial DNA may potentially serve as a mediator to propagate the inflammatory signal in heart failure, as well as other chronic inflammatory conditions, a task formerly thought to be carried out by chemokines. As illustrated here, DNA released in response to heart injury can propagate in this fashion amongst immune cells. Now, as a physician, the question I wanted to ask is, can we stop this? Recently, in a placebo-controlled crossover study, NR was shown to reduce circulating inflammatory cytokine levels in healthy elderly subjects. This is one of the first human studies that demonstrated the anti-inflammatory effect of NR. Consistently, our group enrolled 4 stage D heart failure patients and put them on NR for 6 to 9 days, and saw a dramatic decrease in their PBMC cytokine gene expressions in a pre-post intervention comparison. My working hypothesis at this time was that PBMC mitochondrial DNA-induced mitochondrial DNA release can be attenuated by NR. In the next few slides, I would like to show you some preliminary data that I collected before COVID-19. To test this hypothesis, I isolated PBMCs from healthy volunteers and treated the cells for 4 hours with PCR1. Now, PCR1 is a 421 base pair DNA segment of the circular human mitochondrial genome with high CpG repeats. Since this is a PCR product, all the CpGs are unmethylated. What I observed was that in the PCR1 treatment resulted in about a 40% increase in mitochondrial DNA release, which can be fully attenuated by concurrent treatment of NR. Moreover, the maximal respiration of the cells were severely decreased in the PCR1 or the mitochondrial DNA treated groups, which can also be fully attenuated by NR. What these results suggest is that CpG-rich DNA induces PBMC mitochondrial DNA release and inhibits respiration, which can be attenuated by NR. However, these experiments do not tell us whether the cell, in response to mitochondrial DNA treatment, released naked mitochondrial DNA or the whole mitochondria. So we decided to look at the cells under the microscope. All these cells are stained with TMRM, which is a fluorescent dye that only stains live mitochondria. So these are PBMCs isolated from a healthy 72-year-old male and treated with either vehicle or PCR1. These are 30-second time-lapse videos over a duration of 40 minutes. Let me show you the video for the vehicle-treated cell first. Now you can appreciate live mitochondria inside the cell that's stained red. However, other than that, there's not really a whole lot happening. For the sake of time, I'm just gonna fast forward the video until the end. Okay. Now, for the PCR1 treated cell, I would like for you to focus on the 7 o'clock position where the blue arrow is pointing. Again, you do see live mitochondria stained inside the cell. However, you also see some of these mitochondrial containing vesicles in the background. Okay, now focus on the 7 o'clock position, please. There. I'm not sure if you all saw, but a mitochondria containing vesicle just got exocytosed from the cell at that position. Let me show you, let me play the video one more time. There. Now we repeated the experiment with PBMCs isolated from I think this time a 46-year-old healthy female. Again, I'm gonna play the vehicle video first. Now, unfortunately, the cell didn't stick to the bottom of the glass plate very well and floated out of the frame of view. But overall, there's not a whole lot happening from this particular view. However, in the PCR1 treated cell, right off the bat, you see a lot more of these mitochondria containing vesicles in the background. Okay, please focus on the 11 o'clock position there. So one mitochondria containing vesicle just got exocytosed. And then right there. I'll play the video one more time. There. Now you might ask the question, could this all be a cell type specific phenomenon, since I told you previously that PBMC is non-homogeneous, and maybe by chance that all the PCR1 treated cells happen to belong to one cell type and the vehicle treated cells happen to belong to another. To address this, we isolated monocytes and lymphocytes from the same donor and treated the cells with PCR1. These are some images I borrowed from a hematology textbook. So monocytes are slightly larger than lymphocytes. I think it's estimated to be about 2.4 times larger by volume. Lymphocytes, on the other hand, have this characteristic large centrally located nucleus in the center. Let me play the lymphocyte video first. So again, right off the bat, you see that the mitochondria are more pushed toward the edges of the cells due to the centrally located large nucleus. But otherwise, you don't see a whole lot of vesicle containing, a mitochondria containing vesicle in the background. For the sake of time, I'm just gonna fast forward this to the end. Okay, all right, let's play the video with monocytes that's being treated with PCR1. So as you can see, there are a lot more of these mitochondria containing vesicles in the background. And over time, you start seeing more and more of these vesicles. It's actually a little bit disturbing to look at first. All right. Our modified hypothesis now states that monocyte mitochondrial DNA-induced mitochondria release can be attenuated by NR. Our next experiment would be to see if concurrent treatment of NR would prevent exocytosis of the mitochondria containing vesicles. The data I just showed you are obviously only preliminary, but I thought the findings were very interesting, so I just wanted to share with you here. Next, I would like to move on and talk about 2 human trials involving NR administration in heart failure patients. Drs. Kevin O'Brien and Sophia Earhart spearheaded the first heart failure NR trial, where 30 stage C HFrEF patients with stable class 2 to 3 symptoms were randomized to NR or placebo at a 2-to-1 ratio. The goal of the study was to determine the safety and tolerability of NR in this population of patients. Inclusion criteria include LVEF of 40% or less in TTE immediately prior to enrollment, either ischemic or idiopathic non-ischemic cardiomyopathy as the etiology of their heart failure. Toxin-induced or infiltrative cardiomyopathies were excluded from the study, as were patients who had cardiac hospitalizations or procedures in the preceding 3 months. Now, including the initial drug up-titration period, the study subjects were on the study drug or placebo for a total of 12 weeks, and a target dose of 2 grams of NR per day in the NR group. The primary endpoint of the study was the rate of adverse events. In addition, we also examined explorative endpoints such as functional assessments, 6-minute walk test, and a symptom-based self-reported questionnaire, as well as TTE parameters. Overall, the study drug compliance was very good, over 96% by pill count, and only one study subject ended up withdrawing from the study for personal reasons. Well, first of all, NR was very well tolerated in this population of study subjects. Despite similarities in molecular structure to niacin, NR did not elicit any of the common side effects associated with vitamin B3, including things like generalized pruritus, glucose intolerance, facial flushing, and so on. As shown on the left, there were no severe adverse events in either group. And there were only a handful of non-severe adverse events that did not statistically differ in rate between the groups. The whole blood NAD level was statistically higher in the NR group at week 12, no surprise there, where the whole blood NR level showed a corresponding trend. We did not detect any difference in the 6-minute walk or the symptom-based questionnaire, which is not all that surprising to us since the study was not powered to detect a difference there. Now, our research group recently received an R1 to conduct another human heart failure trial, dubbed NR2. In this study, we're interested to know if oral administration of NR increases the myocardial NAD level, as in the case of animal studies that we went over earlier. We will enroll and randomize 40 stage D heart failure patients undergoing LVAD evaluations to NR or placebo. The subjects will be on the study drug for 4 to 14 days prior to their LVAD surgery, and during which we will acquire a piece of their left ventricular cord tissue. The endpoints of the study are myocardial tissue NAD level and mitochondrial function, as well as the corresponding whole blood NAD level and PBMC respiratory function. In addition, we will be examining the immunometabolic parameters to study the potential anti-inflammatory effect of NR. Specifically, we will be measuring the circulating mito-DEMP levels of study subjects and compare heart failure patients with matching healthy control subjects. In addition, we can also look at the change of circulating DEMP levels in heart failure subjects before and after NR. Here are some mock data reflecting what we expect, or maybe I should say hope to see. So this is not real data, these are things I made up. Ultimately, we would like to construct what I call an immunometabolic relationship, where on one hand, you have heart failure patients with high mito-DEMP levels in their circulation and low PBMC respiration. And on the other end, you have healthy subjects with low mito-DEMP in the circulation and high PBMC respiration. And what we hope to see is by giving NR, we would be able to bring the patients more towards what I call the immunometabolic homeostasis. One question you might be asking right now is why there hasn't been a large double-blind placebo-controlled efficacy trial for NR in heart failure. Well, the short answer is that there's no financial incentive for Chromodex, the company that manufactures NR, to fund such a study. What we are hoping is that if we accumulate enough supportive evidence, including animal studies, observational studies, and focused human trials like this, we may be able to convince the community to ultimately conduct an efficacy trial. To summarize, we went over that NAD depletion is a hallmark of aging. NR and NMN can raise NAD and are safe and well tolerated in healthy and heart failure subjects. Furthermore, boosting NAD is cardioprotective in animal models of cardiomyopathy and can potentially function as an anti-inflammatory in heart failure. And ultimately, and finally, mitochondrial DNA is a potential mediator of inflammatory propagation. It really has been a dream come true for me to finally be able to, quote unquote, treat patients at the bedside while conducting cutting-edge translational research as a physician scientist, as I wrote in the personal statement of my medical school application. I would like to take this opportunity to thank the people who have helped me along the way. First of all, my mentors, Drs. Kevin O'Brien and Rong Tian, who took me under their wings and introduced me to the world of NR. I feel very lucky to have the unique opportunity to learn from two world-class scientists with distinct areas of expertise. I also would like to thank the Tian Lab members, particularly Bo, who showed me the ropes when I first joined the lab and being a true friend. I'm very appreciative of my committee members, Drs. David Dichek, Brock McClellan, and April Stempio-Otero for their guidance, and Dr. Freeman, the UW Cardiology Fellowship, and the division faculty for their excellent training in clinical cardiology. I'm grateful to Sally, Teresa, Heather, Mary, Jessica, and many more who went out of their way to make many things possible for me over the past 4 years. I would like to acknowledge the AHA, ITHS, and the cardiology division for funding. I would also like to take this opportunity to thank the Laughlin family for their generosity and kindness over the years. It truly has been an honor to be the recipient of the Bruce Laughlin Endowed Fellowship. I had the pleasure of meeting John, Carol, Kateri, and Chow last year, and I look forward to seeing you again after the pandemic. Lastly, I would like to thank my wife, who tirelessly stood by me ever since I was a graduate student at UC Irvine, despite her occasional encouragements for me to consider private practice for better pay. I'm just kidding. I would like to thank my parents and my sister, who may be joining us today, for their unconditional love and support all these years. I wouldn't be where I am now without them.